You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech Podcast. I have Toshihiro Shioda, Assistant Professor of Medicine at Harvard Medical School. I'm going to be talking about biology and diseases of the human germline. So, Toshi, thank you for coming. How are you doing?
Fine, thank you. How are you today?
Good. So tell me what is your research involved?
Okay, so we are generating human primordial germ cells, PGCs, which is the earliest precursor of all germline cells, like sperm or like eggs. So every germline cells are coming derived from the PGC, primordial germ cells. And we are making the PGCs out of human iPS cells. You know that Yamanaka got the Nobel Prize by converting, let's say, differentiated cell like skin cells into an iPS cells by deprogramming epigenetically. So what we are doing is we generated iPS cells following Yamanaka protocol, and then converting that iPS cells into PGC-like cells, mainly using a protocol that my friend Jacob Hanna generated. And then we made a modification so that we can generate PGC-like cell culture model out of the conventional iPS cells. The conventional in terms that iPS cells have so-called primed status pluripotency versus naive status pluripotency. The primed status.
What's the difference in those two? Yeah, what's primed versus naive?
Yeah, so it is a important difference. So the primed means it is reflecting pluripotency in vivo resembling the epiblast cells, whereas naive pluripotency is resembling inner cell mass. The difference in vivo is inner cell mass is totally pluripotent, which means it can go differentiate into any lineage of the fetuses, whereas epiblast-like cells, epiblast has a pluripotency which is relatively limited compared to the inner cell mass. And inner cell mass pluripotency is characterized with stronger degree of demethylation and quite different epigenetic profiles compared to the primed status. Unfortunately, sorry.
Quick question here. So just for listeners, because it's getting technical, which is okay, but you're taking skin cells and then you're inducing pluripotency in them, and you're saying there's two kinds of pluripotency: prime and naive, and your goal is to create the progenitors of eggs and sperms from these cells, right?
That's correct. And then,
once you do that, though, what would they be used for? What are some of the possible uses if you do generate them?
Okay, so it's a very good question. So strictly speaking, our research right now is limited towards or directing to generation of sperm or egg. However, I have made an agreement with our medical school and the hospital that we are not trying to go through the production of fertilization-ready sperm or the fertilization-ready egg. We stop before that, which means until the meiosis happens, our research will be stopped before meiosis will be completed, so that we will never accidentally generate fertilization-ready sperm or egg. And then my own research background is I am interested in the environmental effect to the germline. So let's say there are many concerns, such as if you have a very big accident in the atomic power plant, or some toxic materials have been spread, and then you worry about the effect to your next generation, your sons, your daughters, and granddaughters through the germline. And then surprisingly, the study is very, very limited. And most of the data that we know about germline came out of mouse or rat, or those small animals. But recent studies show that the germline biology is quite different between human and those laboratory small traditional animals. And then, therefore, if you really want to know what may happen to humans when they are exposed to bad radiation or the chemicals, one way, of course, you cannot do is expose pregnant female woman and then ask for their fetuses to analyze their germline in the fetuses, which is not possible. And then, therefore, we are trying to generate primordial germ cell model in cell culture. And then we want to know how can we evaluate and eventually prevent bad effects of the environment to the next generations.
So you would be the perfect person to ask this question. So I've thought about this. You know, in humans, sperm are created every 2 days or so, right? Mm-hmm. But in a woman, supposedly in a woman, she is born with most or all of her eggs. So I would think the environmental effects on a woman's eggs would have a lot more time to take effect. You know, forget about exposure to radiation or toxins. What about just lifestyle and diet and stress over 30 years before a woman gives birth, for instance, versus a man going through those same stresses, but every 2 days he has new sperm cells that may be affected very differently than cells that have been around for 30 years. So what are your thoughts there?
That's a great question. Thank you. So then, as you pointed that males can regenerate almost entire germline or spermatogenesis system from the so-called spermatogonial stem cell, SSC. And then, unless SSC is damaged, you can completely recover at least the number of the sperm. Female is a different story. And we have a friend in Japanese university who has done an astonishing work many years ago that for human women, human female patients whose ovary ceased its egg production way before the normal age of menopause. The ovarian follicular cells are sleeping in the ovary, so he took that ovary and then wakened up the eggs with some specific chemical solutions, and then did IVF, and then generated a perfect, normal, healthy, as long as I understand, healthy offspring. And then, so early menopause is actually ongoing, and it is coming to social problems that women who in these days has a career and then don't marry and don't give the first birth until 35 years old, for an example, and then all of a sudden the physician, the doctors tell them that, okay, sorry, but your ovary is depleted. There must be some reason why ovary is experiencing such a problem, and like an early depletion of the functional oocyte. And then it is really an important thing that if you have a male and then if you receive, say, cancer therapy, I'm working in the MGH Center for Cancer Research, so I see many cancer patients. I'm not a physician, but yeah, I can see the patient. And then look, if they are male and then they receive chemotherapy, before that, they may want to squeeze their sperm and freeze it, so that they can get their own children later. Females have a problem. If you are exposed to the very strong chemotherapy and then you want to give a birth to your own child, probably you worry that whether is that safe. I have received chemotherapy and then I lost a lot of hair and then my cancer cells have gone, but my body is damaged. Surprisingly, really, not many information can be given to the patient whether your eggs are still safe or your eggs are quite bad. And then we are doing deep sequencing after irradiating mouse ovary and then sequencing the paps. And then so far, our data suggests that the radiation and the chemotherapy seems safe to the oocyte, but it's only story of a mouse. And then in order to understand how human is affected, we are generating a cell culture model to figure out if there is any method that we can establish to guide human patients whether their eggs are still safe or the choice whether they want to receive those potentially risky therapy or not.
That makes me think of something, and I'm just speculating, but perhaps in a woman's body, the eggs are in a protected state and somewhat isolated from the rest of the body. But then when she goes through menstruation, that, maybe all the eggs are awakened in such a way that they are susceptible to, to more outside influences. So this is just, again, my speculation, but perhaps that's one reason why as a woman ages and she goes through more menstruation cycles, the eggs may have been exposed to more insults or more stress from the environment. So I wonder if there's any correlation between women that, let's say start menstruation late or they for some reason have a longer cycle and less periods, and if that affects their eggs. I don't know. Again, just speculation and thought.
Yeah, so then the fact is, with older age, the chromosomal anomaly of the eggs or the fetuses increase. So that is known. We don't know whether that is because of the accumulated effect of the environment, or is that because just simply prolonged arrest of the egg to in the middle of the meiosis stage is going to cause the chromosomal anomaly. So partly, yes, with old age, your chromosome has a higher risk to get abnormal, say, such as trisomy, those kind of the chromosomal anomaly. But at this moment, it is quite risky to say that it is caused by the accumulated environmental toxic materials. My gut feeling is probably, but without identifying what are those chemical some risks are, and then how can you convincingly prove that it's a very big social impact statement. So it is only my own thought, but it is not very well accepted statement in the field.
Well, okay, so I got a question. Has it been studied, the effect of chemotherapy on women that are in different stages of their cycle? Has that been studied to see what the effects are and how they are different?
So the study field is known as oncofertility. Oncofertility specifically deals with female patients who received chemotherapy and radiation therapy, so cancer therapy, and then how their eggs are safe. And then your question is, surprisingly, does not have a specific answer. So recruiting cycling females and then study whether any chemotherapy has an effect or not is really hard to set up. And so I think that at least what I can say is my hospital, Massachusetts General Hospital, which is one of the Harvard affiliated hospital, the oncofertility chief and I quite intensively talked about that, and then I was surprised that she cannot offer any decent scientific knowledge about whether cycling women has a higher risk by chemotherapy or not. It is not known.
Well, I mean, if you think that, well, I mean, women are still worried, and science still encourages, men. Okay, let me back up. So science encourages men to bank their sperm before they have chemotherapy. So that's saying that an environmental insult, it just happens to be chemical, is gonna cause problems with the sperm. So why wouldn't it do the same thing in women? And depending on the stage they are in their cycle, why wouldn't it have different effects? I mean, it sounds like, I don't know, there's like a fear of even going down that path of thought for some reason. Maybe because people are afraid of going against Darwinism, that it only could be random mutation. But I mean, the whole science of epigenetics itself appears to be because of environmental stress causing changes to the genes, causing epigenetic changes. So why wouldn't that be a very likely or possible mechanism for change?
Yeah, I think that it is actually, we have been publishing papers suggesting that environmental effects are going to cause the germline changes, such as if we expose mouse to tributyltin, which is environmental chemical, which is everywhere in our daily lifestyle, and the pregnant mice were exposed to tributyltin, and then their F1 next generation, which is F1, and next generation, which is F2, and next generation F3, going on, males are tend to prone to get obesity, which means all those male mice whose grandma was exposed to that chemical born normal and looks normal until they are challenged with high-fat diet. But when they encounter high-fat diet, they become obese rapidly, and then their body weight does not goes back to the normal even after we change the diet to the normal chore. And then we have examined their sperm by deep sequencing and epigenetic analysis, and then we see changes in the DNA methylation profile, but we still have a struggle of connecting those observed changes to the phenotype, which is in this case obesity. We can at least say that something bad is happening epigenetically to the germline, but until we show how these changes in the epigenetics exactly causes the observed phenotype, it may be unrelated to different things, and then therefore we are still working on to connect these two things: epigenetic changes we can observe and actual disease, in this case mouse obesity.
Yeah, I had read that the scientist Weissman said there was a barrier between the somatic cells and the germ cells, but it doesn't appear that there is one. It doesn't appear that all the epigenetic marks are wiped out in the germ cell line. It seems like some are preserved. Otherwise, why would you get that effect of mice being obese because of an insult to their grandparents?
So then the Weissman's barrier is a very famous scientific principle, and the fact that we can generate germ cell-like cell line, a PGC, LC, germ cell-like cell culture model out of the IPS is already a violation of the Weissman's barrier, because IPS is definitely somatic cell. And then, therefore, the old days, Weissman's barrier is almost like a dogma. But we are challenging that the germline epigenetic state may be affected through the somatic cell effect of the environment. At least in the nematodes, the worm, the C. elegans, the transgenerational epigenetic inheritance is a known fact. It is not a controversy. It is definitely approved by the community that it happens, and then people are picking up many genes that are directly affecting that transgenerational epigenetic inheritance. The question right now is whether that actually happens in mammals, especially human, and if that is the case, what is the strongest evidence that we can offer? So that is the focus of research of the transgenerational disease inheritance.
So what do you think it's going to take to prove heritable epigenetic changes?
It is a good question. Some people say that it should be DNA methylation, and I don't deny that. And some people say that it can be histone status. And some others are saying that potentially it is not necessarily the sperm, but epididymis, which is the tunnel, the storage place of the sperm, may be affecting sperm to change their epigenetic status. And recently, people recognize that sperm contains histone quite significantly. Probably when I was postdoc, everybody agreed that the sperm does not have histone, and it has only protamine. But today, quite a significant amount of histones are present in sperm, and then therefore it may be inheriting epigenetic information from previous generation to the next. And eggs are always within women's body, so it is very difficult to access, and then the information about the epigenetic inheritance through the female germline is very limited. So probably, so what we are trying to figure out is, other than DNA methylation, is there any changes such as three-dimensional changes in the architecture of the genome, like topologically associated domain, so-called TAD, can be affected by the environmental chemicals, and such effect can be non-specific. It is just deviation from the normal, but it may not go to any specific direction, but it may show a common phenotype like obesity. If energy metabolism is not optimal because of the damage to genome structure, then the consequence may be we always feel hungry and eat more, and so-called thrifty phenotype, that one always feel hungry even though their blood glucose level is very high.
Have we done a longitudinal study on men, sampling their sperm over a period of years and looking for epigenetic changes and genetic changes and other changes in the sperm? I mean, eggs would be harder to do, but we at least done it with sperm.
That area is not my specialty, although I can see that many epidemiologists love to study sperm methylation because sperm is easy to access. And what I can say is that I can see many studies and then many claims such as the changes of DNA methylation, etc. I think that many studies show the changes, but it cannot go beyond that, which means you cannot reproduce the specific change and the specific association to the disease, the mechanistic explanation why those changes causes diseases. That part is largely lacking. So answering your question, I'm not an expert of the epidemiology, so I don't know exactly what is the largest or the best study of the human sperm DNA methylation, but I can at least say that many dozens of studies, many studies have already been done, and then some small suggestions or inconclusive implications were obtained, and then we are still waiting for some definitive answer what exactly is the major cause of the environmental effect to the germline epigenetics.
Okay. So what's your goal with your study of the primordial germline cells? What do you hope to elucidate or figure out in the near term?
So we want to generate in vitro cell culture model of germline. My first goal is we want to know how the environmental chemicals, including the prescription drugs, can affect the germline. There are many drugs. If you buy any drug in the over the counter, it may say that this is not recommended to pregnant female. Talk to your doctor. But I am MD, and if somebody comes to me and then, hey, I want to know what will happen if I take, and if the pregnant woman comes to me, my answer is I don't know. And then I actually called many pharmaceutical companies customer service, and their answer was almost always they don't know. And right now, some misconception is that many drugs, prescription drugs or over-the-counter, says this drug is not recommended to the pregnant females, and only few of them were actually tested for their teratogenicity or other bad effect. The vast majority of the drugs are not even tested. And they're simply saying that, well, we don't test it, we don't know the consequence, and therefore please don't use.
It makes sense to be on the safe side, I guess, yeah.
Sorry?
To be on the safe side, it makes sense to do something like that. But, okay, continue.
Yeah. So in the safe side, it makes sense. But on the other hand, that means that we are just avoiding all those drugs to the pregnant females. Then you encounter the situation that you must use drugs such as cancer. And then, so if there is no other choice but use a drug, we want to know the safety. At least if there is a study showing drug A is more toxic to the germline or human germline than drug B, then we can inform pharmaceutical companies that, okay, looks like A and B are equally effective to the disease, but A is more toxic to the germline, then they can think of developing a better drug and improve the safety. So without any clue, other than just giving that to the mouse, there's no clue whether some drug chemicals are toxic to human germline. So our first goal is trying to make a model to screen the environmental chemicals and major prescription drugs that can be given to pregnant females or the young boys and girls, and then try to establish some sort of the safety criteria of the environmental chemicals and drugs. That's the first goal. The second goal is we want to generate a model of the testicular cancer using the primordial germ cell. And here is a question. Do you know what is the most frequent cancer of young males in the United States?
Young males? Older males, I guess, prostate, but young males, is it testicular cancer?
Yes, yeah, right. So if you are, say, 20 years old, and then you are almost free of cancer except for testicular cancer. So in between 15 to 35 years old males, the most frequent cancer is testicular cancer. Testicular cancer is directly derived from the germline. And studies show strong evidence that the testicular cancers are malignancy of primordial germ cell directly. And then therefore, sorry?
Okay, I'm listening.
Yeah. So, by some reason, primordial germ cells cannot differentiate normally to the germline and stay in the testes for many years, and then eventually form testicular cancer. And half of those testicular cancer are so-called seminomas. Morphologically and marker expression really look like human primordial germ cells themselves. Very similar. And then there are a number of genes that are suggested to be relevant to the testicular cancer carcinogenesis. So we are engineering iPS pluripotent cells with CRISPR-Cas9 and lentivirus vectors to express active oncogene and knock out tumor suppressor genes to mimic human testicular cancer, and then try to figure out if genetic factors can actually generate testicular cancer. The background of this is testicular cancer is a very strange cancer. It is one of the, among human cancers, the testicular cancer's genetic contribution is one of the most strongest. So if your brother has a testicular cancer, the risk that you have a testicular cancer is very, very high. Okay, and then therefore, genetically, it is very strongly affected. However, testicular cancer is shown, very often shows quite local and time-dependent increase in its incidence. Right now, today, in the United States, we are experiencing unexplained strong surge of the testicular cancer in the Latino population. So nobody knows.
No one knows why.
Nobody knows why. And the study came out of the National Cancer Institute (NCI's) epidemiology group, who has been watching testicular cancer incidence for several decades already, and then they published the paper demonstrating that U.S. Latinos, testicular cancer incidence is rapidly increasing, and without any explanation, but it's a fact. So I called the author, Kathleen McLean, and then directly asked her what her thoughts. And then she said that it can be environmental endocrine disruptor, but no strong evidence can support, only because the study is incomplete. And then therefore, epidemiologically, there is very strange increase right now, but nobody can explain. And by the way, in the past 30 years, 3 decades, 30 years, testicular cancer incidence is increasing in almost all of the industrial country, and surprisingly, 1% per year. Every year, incidence increase by 1%. Yeah, which simply means the testicular cancer incidence was effectively doubled in the past century.
That's crazy.
It's crazy. And then many people try to explain that, it's a change in the diagnosis, it's a change in the criteria, pathology, whatsoever. Everything was examined, and then conclusion is, no, there is a real increase. And then therefore, And then therefore, what we want to do is to generate PGC-like cell out of the iPS cells with gene modification to mimic actual cancer, and then see if the genetic factor only can generate cancer, or do we need additional environmental factor to make them really cancer. On the other hand, we are generating iPS cells from testicular cancer, which sounds confusing, but this is another story. We can convert cancer cells into iPS cells. So let's convert testicular cancer cell into iPS, then make PGC-like cell out of those iPS. Those iPS-derived PGCs must contain all cancer-relevant mutations, because originally it was from cancer, so it should contain every cancer-relevant mutations. Then the question is, are these new PGC-like cells already cancer, or they are normal?
Interesting. Okay. I guess that's going to be an interesting experiment.
Yeah. So then that will test whether only genetic factor can cause testicular cancer, or something else is necessary. Anyway, so then
what about looking at you said testicular cancer is on the rise. I've heard this about other ailments. Is it possible for you to find out a survey of 10, 15, 20 different ailments that appear to be on the rise and see if there's any correlation with testicular cancer and with what you're working on? Maybe that would give you a different view of mechanisms or possibilities.
That is, we are trying to generate preliminary data that we can actually do such a study, and then we are going to ask for funding or support for a bigger study involving those epidemiology linked to molecular biology and genomics to figure out. We are not yet at that stage, but that is actually the major goal that we want to go.
Okay.
Yeah, and sorry. So overall, we want to generate a germline model out of the human iPS cells so that we can understand the risk of the germline. What is the risk of cancer? What is the risk of germline depletion? Or what is the risk of the germline anomaly? And also germline mutations.
So we don't have any handle on that versus somatic cells that go through all these issues too. There's no comparison.
So Richard, do you know He Jiankui?
No.
Okay. He Jiankui is the Chinese scientist who claimed that he actually did the gene editing therapy of twin, and then so it was a very big news in the.
Oh, that's right. Okay.
Yeah. Do you know that?
What did he do? He modified them so that they would be born resistant to AIDS. What was the modification that he did?
Yeah, that one. Yes. Okay. So. Technologically, probably he was able to do that, and then again, only an hypothetical technology, someone can generate human fertilization-ready germline out of the skin cells. So the baby from skin cells is technically possible, probably. Not greater than 10 years. I would say the 5 years also technologically it should be possible. But then the big question is that really safe? Because I am handling the human iPS cells every single day. It's very hard cells to maintain. It's a complete normal state. At the beginning of this conversation, we introduced primed versus naive pluripotent stem cell. And then we have recently published with our colleague, Konrad Hochstelinger. Konrad is a corresponding author, and then I did the genome analysis part of that. What we found was human and mouse iPS cells, primed status iPS cells maintain its normal chromosome quite nicely, but naive pluripotency cells rapidly show chromosomal anomaly. It's very rapid, and it is going to be a problem for the safe production of the tissues. And unfortunately, germline cannot be generated from the primed, safer pluripotency. It cannot. Only from the naive pluripotency iPS cells, we can generate primordial PGC-like cell model. And my own study is, I have shown that we can maintain iPS cells in the safe pluripotency, and only 48 hours of conversion into the naive status, we can generate primordial germ cells. Previous studies have shown that you need to maintain cells up to, say, 2 weeks in the naive pluripotency and then convert into the primordial germ cells. But with our protocol, we can maintain PGC-like cells in the safer chromosome status, primed status, and then only minimum length of the time we convert them into the dangerous naive state, and then immediately put them into the PGC. And with that, I think that our system has an advantage to maintain safe and normal chromosome in the PGC-like cells, and that is a major importance of my own cancer study, because if we generate chromosome anomaly as an iPS cell culture artifact, it does not make sense to study the cancer out of them.
What is the difference between a primed and a naive stem cell on the genetic level and the epigenetic level? They have different susceptibility to insult, but what are the other differences?
There are a number of differences. If I go through one by one, I need 3 hours. But and then perhaps the easiest way to explain is the naive pluripotency in vivo, in the real human body. It is only a transient state. Okay, only a few hours, I would not a day even. Several hours, inner cell mass gain naive pluripotency in vivo, and then rapidly differentiate. And then the primed pluripotency, on the other hand, lasts longer, and then the cells proliferate in that particular state. Epigenetically, IMS, the inner cell mass type of the naive pluripotency, is more demethylated than the primed status. This is our hypothesis that too much demethylation of the chromosome is a risk of chromosomal anomaly. So in vivo, in the real life, the risk is limited only for a few hours. But in the cell culture, if we maintain artificially for a long period of time in the naive pluripotency, then we experience as a chromosomal anomaly. And we are trying to figure out if the too much demethylation is a major cause of the chromosomal anomaly in the naive pluripotency. It's an interesting.
What causes a cell to go from naive to primed? Is it cell-to-cell signaling from other cells? Is it just something internal within one cell? I mean, how does it happen?
So naive and primed pluripotency are mutually interchangeable, only changing the cell culture media and growth factor cocktail plus the several chemical inhibitor of protein kinases. So in these days, I think that we have 4I, which means 4 chemical inhibitors, or 5I, 5 different chemical inhibitors, such as GSK3-beta and P38, germ kinase GNK. And so those kinase inhibitor cocktail plus growth factor to maintain the survival of the cells can convert primed status to naive status. And then after the naive status changing back to the primed status media, then they regain the primed status. It is relatively rapid change. And for complete conversion, probably we need 2 weeks. But for my study shows that as short as 48 hours, primed pluripotency can gain germline competence within 48 hours.
Microbiome, quantum computing, virtual reality,
in vivo,
exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now. Companies that are using these technologies
but that only happens when you have
a
sperm and egg.
My goal for you,
I think that inner cell mass is naive, and then it differentiates into primed, which is the epiplasma cell. So that is one way, never comes back again.
Remember, this podcast and its content is informational in nature only.
Is a unique
medical,
set
tax,
of the
legal, financial, or psychological advice
because it is not somatic.
If you enjoyed the podcast, please listen, subscribe, like, and tell your friends about it.
Pluripotency, people say that 3 germ layer, which is ectoderm, endoderm, or mesoderm, but they forget germline because germline is not belonging to any of these 3 layers. So then the true pluripotency is true pluripotency must show differentiation to ectoderm, endoderm, mesoderm, and germline, 4 different cell types. And the germline cells express a lot of pluripotent stem cell markers. And the PGC germ cells, mouse germ cells, can easily reconverted back to the pluripotent state, which is known as embryonal germ cells, e.g., embryonal germ cells. It is another stable pluripotent stem cells.
Okay, interesting.
Yeah. So in terms of the marker expression, the primordial germ cells look like really pluripotent. But as long as the definition of the potency, it is monopotent, which means PGC's only goal is to become germline, not anything else. So it is monopotent. It is committed in vivo.
I guess there's still so much to understand about how this happens and how the priming occurs, and how once the priming happens, how the cell knows which pathway to go along, and
Yeah. I think that so biological interest is how those molecular mechanisms switch prime and the naive and germline like potencies, how epigenetically they are regulated and interchangeable. That is from the true basic biology standpoint, it is interesting. On the other hand, from the industry viewpoint, it is important to figure out how to make safe iPS-derived tissues or cells that can be useful for the human therapeutics.
Okay, understood. So, yeah, I understand why you're making these primordial germline cells, because there's a lot to be learned from them.
Mm-hmm.
Very good.
Yeah, so many people are working on the neurons and the hepatocytes and the pancreas. And then because of my interest in the environment and transgenerational effect, I have chosen to jump into the PGC-like cell culture model.
Well, very good. Well, Toshi, what's the best way for people to find out more? Without blowing their heads up on the complication, but to find out more about your work and your lab, how can they get in contact?
Okay, so I am welcome contact or say my email address. You know that, and then so people can read our paper published in 2017, PNAS, and also people can contact me by email, and that is probably the best way. And I am welcome to talk about how to use say PGCLCs for the monitoring the safety of the germ cell, and especially I'm seeking for an opportunity to talk to the industry people who are interested in the safety measurement of their product. If there is any concern that their product can be used by pregnant women, and they just want to make sure that their product is at least testable or tested for the germline safety, probably we can talk together and then set up some sort of the collaborative work. Now, that would be great.
Okay. Well, very good. Well, Toshi, thank you for coming on the podcast. I really appreciate it.
Yeah, thank you very much for this opportunity.